Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen, the participants of the 8th International Conference on Sustainable Agriculture and Environment. My name is Dia Subositi, and I would like to present our research entitled Study on Genetic Variation of Kelembak, Rheum officinale using RAPD molecular markers. The genus Rheum, a member of Polygonaceae family, consists of about 60 species and is mostly distributed in Central Asia and Asia. The common name for a member of the genus Rheum is a rhubarb. This plant is cultivated for its roots, medicinal properties, and leaf petioles used for culinary purposes. Dried roots and rhizome of Rheum palmatum, Rheum tangucicum, and Rheum officinale are officially listed in the Chinese Pharmacopoeia and European Community Monograph. Chemical contents of rhubarb have identified such as anthraquinone, modine, alemodine, rhein-and-sicones, anthocyanins, flavonoids, and polyphenols. Several Rheum species are used in various traditional medicine globally. The rhizome is usually used as a laxative, stimulant, tonic, and antioxidant. Rheum officinale is a member of the genus Rheum, commonly known as Kelembak. The roots of Kelembak are used as one of the herbal materials for jamu anti-obesity and sought significantly to reduce body weight. Standardization of herbal raw materials is needed to ensure the quality and efficacy of traditional medicines. The standardization of rhubarb faces the challenge of limited germplasm resources. Information on the genetic diversity of Kelembak in Indonesia is limited. The random amplified polymorphic DNA RAPD technique is a popular molecular marker and has been frequently used to study genetic diversity. The objective of this study was to assess the genetic diversity of 9 accessions of Rheum officinale. Plant material for this study was collected from several locations in Central Java Province, among others Candiroto in Temanggung, Mangli Wonosobo, Adipura Magelang, and Tawangmangu Karanganyar. Plant DNA kit isolation from thermoscientific was used to extract the total genome of Rheum officinale accessions. 100 mg fresh leaves used as the samples. The quality and the concentration of isolated DNA were detected by UV spectrophotometer. 6 RAPD primers were used in this reaction PCR. Component of PCR reaction was DNA template, PCR mix, primers, and the nucleus-free water. The amplification reaction program was as follows. The PCR products were checked by using electrophoresis on 2% agarose gel, and the gel was visualized using UV light and photographed by the gel imaging system. Clear and recognizable DNA fragments were denoted as presence or absence of each primer used and in all accessions. Similarity indexes of all accessions were calculated based on dissimilarity. The dendrogram was constructed using unweighted pair group method using arithmetic mean or UPGMA cluster analysis. This data analysis was performed by Entasis software. The results showed that a total of 33 DNA fragments were generated by 6 RAPD selected primers, 14 DNA fragments of which were polymorphic. The percentage of polymorphism DNA fragments ranged from 25 to 50%. Primer OPK3 produced the highest number of the DNA fragments and the lowest number of produced by primer OPK5. The DNA fragments among accessions were used to calculate the similarity index using Dice similarity index. The results showed that the similarity index among accessions were 0.7321, as shown in Table 2. This is the DNA fragment pattern of 9 Rheum officinale accessions using RAPD primers. The dendrogram of 9 Kelembak accessions generated 2 distinct major clusters. The first cluster includes Kelembak Jamu, 3 accessions, and the second cluster consists of Kelembak Jawa, 5 accessions. Kelembak Jamu Mangli was a closer relationship to the cluster of Kelembak Jawa based on the RAPD molecular marker, although the morphology was closely similar to Kelembak Jamu. The dendrogram also revealed that a low genetic diversity of Kelembak accessions. Population genetic diversity is largely influenced by some factors such as breeding system, seed dispersal, and genetic drift. Sample of this research was probably derived from a common source, then transferred and being cultivated in different locations by local farmers. The propagation of rootrop is usually using vegetative propagation by splitting or ground division, thereby generating identical genotype. Kelembak accession in this study were divided into two variants, Kelembak Jamu and Kelembak Jawa, based on the utilization of Kelembak in community. Kelembak Jamu is used as a herbal material for traditional medicine or jamu, then Kelembak Jawa is known as Kelembak rokok cigarettes due to its use for cigarettes production. Kelembak Jamu and Kelembak Jawa can be distinguished based on the morphological characters of their leaves, including leaf shapes, margin shapes, apex shapes, petiole, and root characters such as size and color. Conclusion of our research were: 1. RAPD molecular markers were effective tools for assessing genetic variation of Rheum officinale accessions. 2. The genetic similarity index of Rheum officinale ranged from 74.5 to 100%, indicating a low level of genetic diversity among accession. And the last, the low genetic diversity in the studied Kelembak accession needed more attention for germplasm management. These are the references for the research. Thank you for your attention and wassalamualaikum warahmatullahi wabarakatuh.